Engineering tools for scientific discovery is a broad and important topic, and I think as engineers our bias is that engineering really contributes to the way that we think about how science is accomplished. That is, that science depends a great deal on the technologies that we build to measure, analyze, observe, organize, and predict the outcomes of various experiments. The challenges for us as engineers, of course, lie in making these tools precise and accurate. But consider for a moment the length scales of which we have to make these tools to operate. On a daily basis, we encounter everything from atoms to molecules to complex multicellular organisms, all the way out to the superscales of the cosmos. Each of these present unique challenges in how to consider these tools to measure and observe and to analyze the observations that are made. In considering this particular session as a committee, we aim to really highlight the diversity of scales that are present in which engineers develop tools, but also to highlight the diversity of disciplines that contribute to the development of these tools, from chemical engineering to mechanical engineering to computer science, aerospace engineering, and beyond. It would be impossible for us to cover the complete spectrum of disciplines in engineering and the approaches in engineering that are being taken to consider all of the various problems in science, but that we hope today that you'll enjoy the snapshot that we have assembled here this particular panel of speakers, in which we have tried to cover this landscape of length scales, from direct observations of nanoscale assembly to tracking the interactions and migratory patterns of large creatures on one of our vast frontiers, to trying to understand how another vast frontier has formed, and finally to evaluating how our own man-made systems interact with our natural habitats and whether or not those interactions are ones that would be sustainable for us as a culture. Some particular notes on the session: we'll have 4 talks today. There will be an opportunity at the end of each talk to ask specific questions of the speaker regarding the content of their talk. At the end of the session, we'll have a short panel discussion in which you can ask broader questions that maybe cross some of the disciplinary lines or that tie together broader principles that you have observed in considering this panel. And with that, I would introduce our first speaker of the day, Vinny Manoharan from Harvard University. Vinny is in the School of Engineering and Applied Sciences, and we'll be talking today about some really unique work that his lab is doing to develop new tools to observe how particles at the nanometer scale and micro scale assemble into organized structures.
Start off up front by thanking the people that fund us, NSF, who's funded all this work. So what I'll talk about today is work that was done by my first crew of grad students when I first came to Harvard. They started about 3 years ago. And you'll see their names come up again through this talk to show you what each one of them has done. They've done amazing stuff on holographic microscopy. So as Chris said, I sort of work at the smallest scale of the scales you'll hear about today, the nanoscale. And my lab is broadly interested in this phenomenon of self-assembly. Right? And so by self-assembly, I mean any process, any thermodynamic process, okay, in which you take a bunch of stuff, say molecules, particles, say proteins, amino acids, whatever, and it spontaneously, in solution, due to just random thermal fluctuations, forms an ordered structure. So the simplest example would be just something like a crystal growing out of solution over here. But biology actually uses self-assembly as a manufacturing tool. And as it does amazing things with self-assembly to make incredibly complex structures. So I'll show you just one example over here. This is called a Clathrin cage, all right? And this image over here is an electron micrograph with atomic resolution. And everything you see here, all the features of this are self-assembled. Your body at every moment is synthesizing proteins which have some amino acid structure, and they come out, the Clathrin monomers come out, they self-assemble into secondary structures, tertiary structures, quaternary structures, even higher order structures, all the way from the angstrom scale self-assembling all the way up to tens of nanometer scale. Okay? And this happens spontaneously without any energy input. And it happens robustly also, because these things are involved in cellular transport, so it has to happen robustly. If things don't happen robustly, then catastrophic things ensue. All right, so people who work in nanotechnology, people who work in material science like me, look at things like this and say, wow, that's amazing. I would love to be able to do the same kind of thing, assemble complex functional structures without doing any work at all. It's a great way to manufacture things, right? I would love to be able to do this kind of thing, where maybe not using amino acids or proteins, but take some soup of building blocks that a chemist synthesizes, throw it into solution, and have it come out to be a functional product. So, for example, throw everything into a pot and have it come out to be your latest, say, Intel microchip. This is the dream, right? We look at this and say, this is what we want to do. Biology does it. Why can't we do it? Well, it seems a little bit far-fetched. People are doing things, though, these days, even the electronics industry with self-assembly. This is an example from Hitachi, Paul Neely's group, and other collaborators at MIT. They're using things called block copolymers, all right, which are synthesized at nanometer scale type objects, and these things can form these ordered structures spontaneously. They're using these as patterned substrates for hard drives. So they can achieve a higher density by having a patterned substrate. This is a very easy way to get a patterned substrate with the scale of nanometers. But it's still a bit of a far cry from making that Intel microchip. And so what I want to focus on then is what is it that we really have to do in order to make that microchip, in order to make self-assembly work for us. And the truth is, we don't really have a good understanding these days of what it's going to take, what kind of building blocks, what kinds of interactions we need in order to get self-assembly to assemble the things we want, the complex structures that biology makes that are functional. And as George Whitesides pointed out a few years ago in a review article, what we have at the moment, and we still would, I would say this is still the case, is a fairly simple set of structures that we can assemble through self-assembly. So how do we get beyond that? What's the question here? Does it have to do with us having to do better chemistry? I would say the answer is actually that we have to be able to do the physics. We have to understand physics. The problem with self-assembly is actually a physics problem. And it's here, complex systems, right, where I think the key questions are. We don't know enough about how interactions guide the formation of complex structures. And so everybody is willing to give nature credit for being a good engineer, right? Nature's an amazing engineer. But I think people don't give nature enough credit for being a good condensed matter physicist. All right? And so it turns out that the problems we're talking about here, the problems of self-assembly, how do you self-assemble something? How do you get it to do so robustly? How does biology do it? They're all related to problems that condensed matter physicists have been looking at for a long time. Right? And this was pointed out in an article by Phil Anderson, 30 years ago, essentially, where he said that, look, even if you understand how to synthesize something which has some defined interactions, even if you understand the interactions between two particles, it doesn't tell you anything unless you actually go through and do the experiment and do the work to do it about how a whole bunch of these things will behave. It doesn't tell you about the collective phenomenon. It's related, but it's a far cry from understanding the interactions at some scale to understanding how global structure arises. Okay? And this is what he's meant by when he said more is different. More than two, things get complicated very, very quickly. And so all of these problems we think are related, right? How does biology assemble structures so robustly? How do we engineer self-assembling systems? And all of these questions that condensed matter physicists have been looking at for a long time. Things about why is it that sometimes liquids don't form crystals, they get trapped into metastable states like glasses? Why is it that other times they form crystals readily? And even questions related to all sorts of other phase transitions in condensed matter. And so my point of view on this is that to really make progress in the field of engineering things through self-assembly, we have to understand the physics of complex systems. So it turns out that in the last 10 years, a lot of what we've discovered about the physics of complex systems has come from, not from biology, but from actually a different place. It's come from microscopy, Microscopy has been the tool, but not electron microscopy and all these tools that have been done on biological systems. What we understand now about self-assembly, a lot of what we understand, and condensed matter physics, comes from, in the past 10 years, studies of these things, which are model systems, okay? Systems which are actually larger than the biological scale. Right? So systems like colloidal suspension, suspensions of a bunch of nanoparticles, say like half a micron now in scale, small enough that they still exhibit thermal fluctuations and can self-organize and form phases and everything, but large enough that you can actually see them in 3D. And you can see them using the main tool, which is the confocal microscope. And so the confocal microscope, what it is it just scans a laser mechanically throughout an entire sample and builds up a 3D image by scanning a laser throughout. And so you can take a confocal microscope, you can look at one of these model systems, which is chemically quite different from the systems at the atomic scale, right? But physically encodes the same kinds of ideas. You have multiple interacting particles. In a sense, it's a very simple model system for understanding complex systems. The simplest complex system, you might say. And you could build up 3D images of this, which contain not only structure, but also dynamical information. And so this is an example of one of the 3D images that people have built up over here. And then this is actual experimental data now on one of these colloidal systems, model systems, where you see this is a computer rendering of the experimental data. You can see all the fluctuations of all the particles at once. And with this information, you have essentially everything you need to understand the thermodynamics of a process going on. So this example here, what they're showing is a liquid that has become a glass. It's become trapped in one of these metastable states. It has failed to self-assemble into a crystal. And then people have looked at all sorts of things of phenomenon using this tool related to material science, related to self-assembly, related to nanotechnology. They've looked at melting at grain boundaries, so they made these model systems that crystallize and then they melt them and they can see exactly how things melt. People have looked at gelation. Again, this is all experimental data. It's very nicely colored and rendered, but this is actual data from the experiment that's been rendered on a computer. We can look at the real space structures of gel, see exactly what's going on and the dynamics in there. You can even look at defect propagation, dislocations in crystals, all sorts of things. So this tool has showed us a lot about the dynamics of complex systems. But the problem is, for going further, we're at a bottleneck now. And the issue is that in all of these cases, the dynamics that are being probed are actually quite slow. And in fact, in this case here, this is a glass, so it happens to be a system that moves about very slowly. And the time between frames here is actually a minute, so this is quite sped up quite a bit. To actually understand some of the processes I want to understand about self-assembly, I want to be looking at timescales much shorter than that. And confocal microscopy is not a way to do it. So confocal microscopy is just too slow for that. And it's slow because you're limited by the time it takes you to mechanically scan through the entire sample with your laser. And at a minimum, if you want to look over a bulk sample, that takes about a second. Okay. It also happens to be a very expensive instrument. Confocal microscopes have now become a central facility item for universities. You know, they're $500,000 to $1 million, and they're huge, bulky items, which makes doing the kind of science that we want to do, which is small science, quick, dirty experiments, quite difficult because you have to set up everything on this central facility. So what we want is to be able to image in 3D, like confocal, with a large depth of field, large compared to the particle size, so I can see everything that's going on in bulk, right? So again, like confocal, but with an acquisition time that's much, much smaller than the smallest time scale I have in my system. So I'm capturing all the information I want about dynamics. So that time scale, I want to get down to millisecond type timescales, 3 orders of magnitude faster than confocal. And because I'm perpetually broke, I want to do this very cheap. And I want to do it with a simple instrument that I can make a lot of in my lab. So it turns out that there is actually a very simple way to get an instrument that does 3D imaging with all of these things. How simple? Well, it's as simple as taking a laser, and shooting it at your sample. Okay? When we first started working on holography, I had no idea that holography, it's something that sounds so complicated, would be as simple as shooting a laser at something. So this is the kind of experiment we did. You take your sample, this would be your colloidal sample, which you want to see self-assemble. You shoot a laser at it, and then you have an objective over here. And the important thing is that the laser is a coherent light source, so it interferes with itself. And on a camera over here, you record the interference pattern. This is the basis of the experiment. It's the simplest experiment you can do. How does this work to give you a 3D image? So I'll explain it with this little animation over here. Let's say I have a particle, a point particle, for argument's sake, and I come in with a plane wave, coherent plane wave, laser light. Now, the particle will scatter some of that light, okay? But it's not going to scatter all of it. So what I'll have over here is two waves. One is the incident plane wave, the undiffracted, if you like, part of that plane wave. And the other is the diffracted or scattered wave from the particle, which we'll say for a small particle is approximately something like a spherical wave. So now, if I put a detector over here, what I will see is an interference pattern. And I'll see an interference pattern between the scattered wave and the transmitted wave. And the interference pattern has fringes, and the reason it has fringes is because the phase difference between these two waves changes as a function of position, because one of them is curved and one of them is a plane wave. If I take a picture of that, say on a piece of photographic film, it might look something like this. Okay? And these fringes that you see here, these circular fringes, encode, I claim, all the information about the 3D location of this particle in this 2D image. The center of these encodes the position of the particle in the plane parallel to the detector over here, right? And the distance between fringes encodes the position of the particle with respect to the detector, so along the propagation direction. And the reason for that is that the distance between these guys varies as you get further away because the curvature of the wavefronts changes. Okay? So this 2D image contains all the 3D information about that particle. And in fact, there's a very easy way to recover that information. To recover that information, you can do something as simple as you take the pattern over here, you put it on a film, you make an amplitude mask of that pattern. Okay? So I have an amplitude mask, and it's dark wherever I had destructive interference, say, in my fringe pattern. And then I shine light back through that. And if I shine light back through that piece of photographic film, the whole film acts as a diffraction grating. It acts as a lens. And what you recover is, in fact, a real image of your particle, exactly where it was before when you did the experiment. And the reason you get it over here is because you've blocked all the light that would have arrived here out of phase. So you only get a coherent superposition. Okay, so this is a way to do fast 3D imaging, and so we call it digital holographic microscopy. Digital because nowadays we don't do this on a film camera, we do this on a digital camera, and we numerically propagate light back through the sample. It's based on what Gabor did in the 1940s, except using a digital camera instead of film. But it represents a way to get fast 3D imaging. So here's an example. This is data that we take from a particle that's just floating around in solution. Okay, it's a half second of data. There's 400 frames in there. It's played back at real time. So it's a very small, short movie here, right? It doesn't show too much. But by reconstructing it in this way that I just showed, you could take this data here and slow it down and generate, actually, the position of the particles on time scales of 2 milliseconds or so. So now I've slowed it down. This is a 1 micron box. You can see the particle move around in all 3 dimensions, and you're tracking it to a precision of 10 nanometer displacements. So this is a quantitative technique. And in fact, if I look at a quantitative measure of that particle's displacement, the mean squared displacement, I can measure that down to timescales of 2 milliseconds or so. Einstein told us that if I measure this quantity, the mean squared displacement, it's linear in time. The proportionality constant is the diffusion constant. If I do this experiment, I see that in all 3 dimensions, I get a result that agrees pretty well with the expected diffusion coefficient from theory. So it's a quantitative technique. And again, I want to just point out that it's linear down, this functional form is linear down to about millisecond timescales with nanometer scale displacement. So quantitative down to the kind of precision that we want for these kinds of experiments. All right, so what about self-assembly? So here's what we like to do in my lab is create systems that, as I said, aren't chemically equivalent to biological systems, but which encapsulate the fundamental physics. This is one such system. Particles which are trapped on the surface of a droplet. We like to look at, if I have a bunch of particles and they're on the surface of a droplet, how do they interact to form a structure? And it bears some similarity to viruses. If you have a bunch of proteins, they'll tend to form icosahedral structures. Our systems tend to do the same thing. We want to be able to look at this the system in real space and in real time and see everything it's doing at once. This is impossible to do with a confocal microscope. And the reason is that, this is a picture of the thing under a bright field microscope, right? And you see things going in and out of focus all the time. At the same time, the droplet's going to be moving around, it's rotating, it's translating. We're looking at a bulk system. So we have to have a way to see everything that's going on at once. And in a confocal, by the time you've scanned through the droplet, it's moved out of the field of view. So everything gets blurred out. With the holographic microscope, though, you can take advantage again of another feature of holography, which is that I've shown you what happens when you have one particle in the system, right? When you have many particles in the system, it turns out that the hologram is very easy to imagine. It's just the linear superposition of the holograms from all the component particles. Right? So you get a pattern that looks like this. It's basically adding up all of these guys linearly to make the full hologram. And it turns out it's very easy to extract the data from that. What one does is one just does a Fourier transform. You know how individual particles or points scatter. This is the hologram of the whole system. If I do a Fourier transform, I can do a convolution between these two, and I can look where that convolution is maximized. That tells me where my particles are. So from a single image, you can build up a 3D image of all the particles in the system. You do this, you get a movie like this. This is, again, actual experimental data. So this is now looking at all the fluctuations of the same system I just showed you before, 10 particles sitting on a droplet, and all the fluctuations going on at the same time. Okay, and these are the projections on the coordinate axis over here. And this is real time now. This is real time, played back 30 frames per second.
Can I ask a question about the previous slide?
Yeah.
So it seems like at a particular depth, right, because you'll have that will change the spacing of the rings.
Yes.
So if you made a convolutional mask based on it, wouldn't that only be effective at one depth?
Yes, that's right. So you have to convolve, you have to make your impulse response at multiple depths and then convolve simultaneously. And I'll talk about that in 1 second. Thank you. So again, this is a quantitative technique, and the nice part about doing this is that you get essentially all the information about the statistical physics of the system, right? So I can go through now, and from the positions and all the particle positions, I can go through and calculate energies actually, and I know that through the Boltzmann distribution. So I'm actually doing a thermodynamic measurement. And you can go through, and we can actually go through all the configurations that this system went through, all the statistical mechanical microstates, and bin them all by energy, and we can do things like extract, for example, ground states of the system, right? And so this is one of these extracted ground states, what we believe to be the lowest energy configuration of the system. This tells us something about how the system self-assembles, right? And that's the point, right, of why you want to do fast 3D imaging, because it gives you all the statistical mechanical information about the system. It tells you everything thermodynamically in principle. The challenge is, of course, there's always a catch here, right? You get all this information because you have this fast acquisition time, and we can start to understand the statistical physics of self-assembly. But the challenge is that, it's a huge amount of data. So just the raw data we're generating, and this is a theme you're going to be seeing actually throughout this whole session, I think. The raw data we're generating is about 1 gigabyte per second. Okay, just of the movies, of the holograms. And then, as someone pointed out, we have this problem of reconstructing this. We have to reconstruct it at multiple depths simultaneously through these Fourier transforms. We typically do 1,000 of those per frame, so you're generating about 1 terabyte of image data, volumetric image data, per second of actual acquisition data. Right? And it takes a long time actually to process that. Even on a typical CPU, it's about, you can do maybe order of a few Fourier transforms per second on a megapixel image to process that data that I showed you before, which is a second or a few seconds of data. It takes about a week. So a CPU week to process a second of data. Now holography's been around for a long time, right? But really the reason that we're using it today is because this is now something that's manageable. Right? I can handle waiting for a week for data. And I can especially handle this, I can deal with this, throwing this at the computer, if it allows us to do experiments that we just couldn't do otherwise. And this is the idea here. So we think we can do a little bit better. We think we can actually now show that we can measure interactions without actually doing reconstruction. And so what we do is we shine a laser at a bulk colloidal suspension. We generate a hologram. There'll be some interacting particles in there, okay? And we want to back out the interaction between those particles. Well, one thing you find from these holograms is that, heuristically, you can kind of extract that information just by looking at it. All right? If I have one particle, it looks like this. If I have two particles, it's still you have this circular fringe pattern, but then you also get this linear fringe pattern, and it's just like a two-slit diffraction pattern, right? That linear fringe pattern encodes information about the particle separation in 3D. So the distance between them and the ratio of intensities between them gives you the orientation. So what we do now is we actually fit these holograms. Instead of reconstructing it, we fit it to an exact scattering calculation from spheres, right? Because in the end, we're only interested in the positions of these spheres. I don't have to go through this whole process where I generate the whole 3D volume. Right? So if you can fit it, this works out a lot better. And in fact, so we fit it to an exact solution. That has the advantage of it saves us an enormous amount of space involved in the data. It is also extremely precise. So this is a picture of the center of mass of this doublet particle as it's rotating around. It's moving over 10 microns or so. Even so, even while it's moving there, we can detect this vibrational distance between it to a precision of 5 nanometers, even as it's moving back around. And that's because we're using an exact scattering model for that. And so from that, you can get the separation distances, and again, you can get the interactions. And my dream is, and this is still an open question, is it going to be possible to do this kind of problem, fitting this data set for many, many, many particles simultaneously? And I don't know what the limitations of that are, but this is a good conference to kind of discuss things like that. All right, so the last thing I will briefly talk about is now getting back to biology. We were also interested in studying dynamics of cells. I brought this whole thing started with living systems, right? So what can we do with living systems with holography? We're really interested in looking at, say, lots of different cells and how they organize, stem cells, cancer cells, et cetera. This is a new project in my lab, and so the conventional way biologists do an experiment like this is they'll take their $500,000 microscope, right? And they'll grow some cells in an incubator, but then they'll transfer that incubator over to the microscope. They'll build this fancy, expensive environmental chamber around the microscope, and they'll do the experiment there. Now, I don't have the resources or the time to do an experiment like that. Looking at this, it seems a little bit ridiculous. You've made a $100,000 microscope or a $500,000 microscope into an incubator for cells. Why can't you just take like a cheap microscope and stick it in here? That was the question that I wanted to figure out. And so we had an undergrad intern in the lab, very talented undergrad from University of Chicago. And so what I did is just said, look, I gave her a credit card and an account on eBay. And I said, build me a cheap microscope that I can use to image cells. And so she came up with one. So what she came up with is this. You take a digital camera, DSLR camera you get on eBay, you use that to image your hologram. You'll have a lens and some assorted optics in here. And then you just have a laser diode, which you can get from any electronics parts manufacturer, and a little microcontroller, a $20 hobbyist microcontroller that you hook up to a 9-volt battery and which controls the whole experiment. The whole setup costs a few $100s, a little bit more with all the optics involved, and you could program it all. And the nice part is it's all self-contained, it's all battery powered, and in principle it's portable as well. How well do you do with it? Well, you actually can do pretty well with today's consumer electronics. So this is a hologram, okay, of a resolution grid, and we can reconstruct this. This is generated from this portable microscope, and you can see down to less than 3 micron resolution. You're resolving less than 3 micron fringes at about 800 nanometer wavelength. And you could do that over a huge depth of field and with a very little amount of light, 100 microseconds exposure time. Can you image cells? Yes, so we've just started imaging cells. This is a hologram of some cheek cells. This means that you take a Q-tip and you swab your cheek and you put it on a slide. You're essentially spitting on a slide. And then you can reconstruct this, and in fact, if you go through, you can actually optically section these cells, and you can go through and you can we're actually able to see some subcellular features. These are fairly large cells, but you can see, for example, the nucleus of the cell over here as well, all for less than $1,000. And so really, thinking about this, what makes this all possible is, and I've alluded to this many times, is the advances in semiconductor technology. This is what makes all of this experiments possible. On one hand, you have the advances in sensor technology. Even digital cameras now have the resolution to be able to see all of these interference fringes and construct high-resolution holograms. You have high sensitivity, high frame rate was the other thing I talked about. Laser diodes, we no longer are limited to these gigantic lasers when we want to do an experiment. We can now just use an off-the-shelf diode and and hook it up to a small power source, a battery, and of course the computers. The computers are really what allow us to separate acquisition from processing, right? And that's what allows us to get to acquisition time. Processing is still the bottleneck, even without Reconstruction. This is a challenge we have to face, but that's going to get better. And as I said, I'm okay with that if it makes it possible to do new experiments. And I think that's the key upshot here. You can do things now with these kinds of advances that were simply not possible to measure or to see before. And that leads to a whole new kind of role for the microscope in the lab, not as a central facility, but really as an add-on to little individual apparatuses here and there so that we can use it more effectively as a tool for scientific discovery. So with that, let me thank the students who are involved with this and our collaborators at MIT, George Barbastadas, who really got me started on holography, and NSF for funding this, and thank you.
We have time for a couple of questions. Loren, do you want to help me run the mic? So it would be great if you could speak into a mic. Give your name. Please state your name and the title where you're from. I saw Robbie's hand first, so.
Hi, Robbie King from RPI. First of all, just beautiful talks. I had a question about the scattering from multiple particles. Basically, is the interference pattern unique? And if it's not, will that be a problem when you have a large number of polydispersed particles or the shapes are not spheres?
Okay, yeah, so good question. So is it unique? There are degeneracies, right? So, you can't tell if it's here or if it's here, right? But that's where the time comes in. If I can track things as they're moving around, then I'm not sampling random things. I can see what each particle is doing. The polydispersity issue is another thing. So we fit not only for this separation, but we actually fit for 9 different parameters, in fact. You can fit for not only the separation, but the individual sizes of each sphere and their refractive indices as well. So you get all of this information from the fit. There's a tremendous amount of information contained in that hologram. Yeah.
Lucas Novotny, University of Rochester. I very much enjoyed your presentation of the holographic microscopy, and you listed the pros and cons, and it appeared to me that there must also be a signal-to-noise trade-off, because, say, in confocal microscopy, you concentrate all the scattered light onto one pixel, whereas here you spread it out over many pixels, so that must be, make your signal-to-noise a little bit worse compared to confocal microscopy.
Yeah, absolutely. But I would point this out. So typically in confocal microscopy, what people do is not scattering, but fluorescence, right? And so, in fact, we're actually much better in terms of signal-to-noise than fluorescence. So all the experiments that I showed you with the tracking particles with confocal, those are all done using dyed particles. You could do it with scattering, but in order to get things index matched and transparent, you get very weak scattering. So in fact, signal to noise is better because we're relying on scattering rather than fluorescence. There are still issues that are going to pop up, right? But the nice part is, well, 2 things. One is that the laser, you can come in with a pulsed laser, and this is what we do nowadays. We can come in with a pulsed laser, deliver a lot of power in a short amount of time so that the total dosage to the sample, integrated dosage, is small, right? So that helps us. And the other thing you can do, which I didn't show here, but the undergrad designed this cool trick for the portable microscope. You can sort of dynamically extend your dynamic range by taking 2 exposures, right, at different laser powers. So you can capture things that weakly scatter with a high laser power exposure and things that strongly scatter with a low laser power exposure. So there's tricks like this that you can use to address some of these signal to noise considerations.
May I ask a related question? Is there also a limitation of how dense you can pack the particles? Because probably that holographic reconstruction doesn't take multiple scattering into account.
No, well, in the case that I showed you with the fitting, that does take into account the multiple scattering. But you're absolutely right, and I briefly pointed this out on the sort of cons of this. How many particles can we actually study? And this relates more to your signal and noise question. The information, I mean, you're getting a 3D image from this 2D projection. The information contained in there is some combination of the resolution, the pixel resolution of that camera, times the dynamic range. At some point, right, I'm superposing all these holograms together, and at some point the differences between them have to be between my signal and noise, and I can no longer see my fringes. I don't actually know where that limitation lies. I suspect that as of now, we could maybe do 100 particles. We're not going to get to 1,000 without significantly extending that dynamic range, but that's absolutely the main limitation in terms of, the other big limitation in terms of doing experiments with comparison to confocal.
Andy Molish from University of Southern California. Thank you very much for this very interesting talk. There seem to be some techniques actually in wireless communication engineering related to the analysis of scatterer locations based on multiple antenna images. And therefore the processing, actually a Fourier transformation, is not that popular anymore, but rather maximum likelihood techniques that don't suffer from noise enhancement that much. I was wondering whether anything similar has been explored in the holographic imaging area.
I'm not sure, actually, but this is a great point. This is why it's good to be at this meeting. So, I mean, we should talk more about this because, we haven't, my lab does not do extensive signal processing work, right? We do whatever is the simplest thing. There are other people like George Barbastatis who works on these kinds of problems, but I've never seen anybody apply these kinds of technologies to actually doing, science like this, right? So we've seen things that are done in principle, right, in optics journals, et cetera, but actually using some of these techniques might be, for fitting the data, right, maximum likelihood estimation instead of Fourier transform techniques might actually, give us significant progress in a short time. So, I'd love to hear more about that.
Okay, let's thank Vinny once more.